Hello everyone, welcome to today's live broadcast, Deep Sequencing of the First 10,000 Human Genomes. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals. Before we start, there are a few instructions. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, and to get your credits, click on the CE button located in the bottom left-hand corner of your web page. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see the presentation properly, let us know by clicking on the support button at the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome Amalio Telenti, PhD. Dr. Telenti is the head of genomics at Human Longevity Incorporated and a research scientist at the J. Craig Venter Institute. He directs the team working on genetics and genomics. Recently, Dr. Telenti led the analysis of the first 10,000 genomes sequenced at HLI. He trained in internal medicine and infectious diseases at the Mayo Clinic in Rochester, Minnesota, in microbiology and genetics at the University of Bern, Switzerland, at the Albert Einstein College of Medicine in Bronx, New York, and at the Reagan Institute of MGH, MIT, and Harvard in Boston. Between 2007 and 2014, he was a professor and director of the Institute of Microbiology at the University of Lausanne, Switzerland. Dr. Telenti has published extensively and received many national and international awards, including the prestigious Siglecl Award, one of the highest distinctions in medicine in Switzerland. In 2012, he was elected as a member of the Swiss Academy of Medical Sciences. He maintains academic affiliations with the J. Craig Venter Institute and with the Department of Medicine at the University of California, San Diego. I will now turn it over to Dr. Telenti for his presentation.
Good morning, and thanks, Judy, for the introduction. It is a pleasure to present some of my work, both at the J. Craig Venter Institute, an academic institution, and also at Human Longevity, which is a corporate institution working on genomics. Today, we are going to discuss 4 topics. One is the concept of what do we mean when we refer to sequencing a genome. The second one is the type of dimensions and aspects that can be learned from studying a large number of genomes, in this case 10,000 genomes, at what we call a deep sequencing level. Some short aspects on interpreting data and some examples, and then as an application, I will show the impact of rare human variation on genetics of response to viral infections. And here I mean things like a flu or other very common infections in the population. So on the first topic, what's a genome? I give you here the numbers from the last 18 months of publications of large-scale efforts to sequence human genomes. On the top is the very well-established, very well-known 1000 Genomes project that was published in 2015 as one of the most recent publications. And then you see that we have other countries, we have the African genome, and overall, the total of published genomes approaches 14,000 genomes in the public domain. Now, you have to pay attention to the third column called average coverage. This is how many times, in average, we have read this code. It's like if you are reading a book and you are asking how many times you go through the book to make sure you got it right and you memorized and you did not miss a word or a chapter. And you see there that the average coverage varies profoundly between some of these studies. So some people read the book 4 times, like in Sardinian Genomes. Some people read the book 32 times, like in the Japan Genome. And you will see in a moment that this makes an important difference. And then finally, the last column talks about number of deep coverage. And here we are referring to those that have higher than 20x coverage in these published genomes. And there you see that actually the number decreases to 2400. So when we are referring to a genome, one of the important things is to understand what is being referred to in terms of depth, and depth in sequencing the genome quickly translates into quality, precision, false positives, false negatives, false discovery rate. So when we cover the genome at great depth, we obtain what you see in the upper part of this slide. These are all the autosomal genomes, and you can see that I place a blue color there where the deep sequencing of genome reaches. And you see that actually, contrary to the belief of some people, we never sequence the genome completely except if we make a major effort and use multiple techniques. When you just use the most commonly available technique, that is a short read technique like the one used by Illumina, you will have a difficulty to go through all the complex parts of the genomes, and those include, like you can see here, for example, in genome 14, I'm going to paint it out. So you can see here, these 2 genomes have a complete lack of sequencing on the telomeric part, and then we can put another genome part, like here, where typically you are looking at the centromere is not easily sequenced across any one of the genomes. So you can see most of the chromosomes have gray areas around the centromere. This is known because of repetitive sequences, because of what we call low complexity sequences. So the first lesson is that you have to read the genome multiple times for precision, and also you have to read the genome at great depth to try to cover the most of the genome territory. I put on the bottom what is the equivalent for exome. So when people say we have sequenced the exome, they are sequencing, of course, 2%, 1.5 to 2% of the genome that corresponds to the coding. And you can see the amount of information that is not being recorded by exome projects. I don't put in this slide aspects such as genotyping arrays, like used in GWAS analysis, or like used in some commercial companies that offer direct to consumer, because genotyping arrays only analyze unique positions between 500,000 and through a technique called imputation, several million, and of course they will not be even represented at this high scale. So to summarize this, sequencing of genome requires for maximal precision and maximal coverage of the genome requires extensive depth, and this has not been consistently reached by many of the exome projects. And anything that is not a full genome coverage, of course, as you can see here, gives a minor completion of the genome sequencing, even though, for example, sequencing the exome can be extremely informative on purely protein coding areas. When we sequence these very large numbers of genomes and we go all the depth, of course, what we are seeing is the true nature of human variation. And what we obtain quickly is this type of graphic where to the left, you can see many millions, in this case, 80 million variants that have seen only once. So think and reflect about this. We are looking at 10,000 people, and we already have 80 million of variants only since once. We have variants since only twice, 3 times, 4 times, etc. And only all the way to the right, what it says more than 5%, and I'm going to put an arrow right here. This is what is called common human variation, more than 5% allelic frequency in a population. Basically, when you are genotyping with an array, when you are genotyping in a direct-to-consumer, you are looking only to the right two columns of this graphic, the 5%, possibly the 1% to 5%, and if you do imputation, that means deducing what is the other SNPs present around, you can reach the other arrow, that means 1 to 0.1% of the population. So if you think about this, what we call common variants are actually rare variants. And what you call rare variants, the ones that are only seen once, are extremely common and probably very important for the future of discovery in human genetics. So let's summarize a bit to give you a scale of the task ahead, what the genetics and genomics and medical community will face as fantastic challenge in the next coming years. I have represented here the amount of variation that I have observed going through now 20,000 genomes at human longevity. And this is the large black circle. I have observed 400 million variants, mostly because of variation. The next circle, the red circle, is the content today of the public database DBSNP that you probably use, that contains 184 million variants. So it tells you that already just by sequencing at great depth 20,000 genomes, we are basically already doubling the amount of knowledge available in the public database. Now, what is very impressive is when you start getting to the center of the slide, is that that blue circle is all the coding variants known today. And these are deposited in EXACT at the Broad Institute. And you can almost say that the whole planet is today turning around inside this blue circle. That means we have a tremendous interest, and that's appropriate, on the coding diversity in humans. But you also need to appreciate that that's only part of the picture of human diversity, because it dwarfs to see this exact data in the midst of what has been already observed in the non-coding part of the human genome. And there is still more you may not see, but there is a pixel right in the middle of the blue circle that you may not actually even see. That is ClinVar. ClinVar is the NIH-supported repository of clinical variants, and today contains around 120,000 variants. It's a very important tool, but again, we have to put it in the perspective of EXACT. We have to put it in the perspective of all the human variations that we are currently identifying. And even behind that pixel, there is something that is probably even less than a pixel, is that even within ClinVar, there are sets of variants that are better known and better studied for Mendelian disorders. That's probably around 10 to 15,000 variants are really carrying confidence signals with agreement between laboratories. And then there is something coming from the GWAS that is statistically confirmed, even though usually of a modest individual impact, that is the GWAS variants, and those are 16,000 also coming from public repositories. So recapping on this figure, we are making tremendous progress, either through GWAS associations with common disorders, common traits, either from Mendelian. There is a huge effort in the community to try to solidify this knowledge into ClinVar. There is a huge effort trying to cover human variation in coding regions, led by EXACT. There is the main effort of recording all these variants in public databases like DBSMP, but we are expanding the amount of human variation extremely rapidly, and this is going to be a very important challenge for the community to understand their meaning in human diversity, traits, and diseases. Where are all these variants coming from? There are many mechanisms of having so many rare variants. There is founder effects for communities, there is ancestries, populations, geographical variants, all the things that you can imagine through human evolution, migration, and all the environmental selective pressures that may lead to selecting and then eventually fixing variations. There is one mechanism that is important, is de novo mutations. And de novo mutations typically look like in this graphic, where we see a child with a neurofibromatosis. This is a disorder, a rare disorder. And you can see a variant in green that is more or less 50% of the BAM files of the reads. Those are the gray areas in the square. You see that the mother doesn't have that green variant. You see that the father does not have that green variant. That typically is a de novo mutation acquired by two main mechanisms. One is, in this case, very early post-zygotic mutation of the embryo. The other possibility is that this variant exists as extremely minority variant in the mother or in the father in a somatic form. That means that some cells in the body may carry this mutation, and it may be transmitted to the embryo during the conception. So this is an important mechanism, and probably any one of us carries 50 to 100 de novo mutations when we are born, and we're different from our parents. So this is an important motor of generating human diversity, human rare variants, and then it is evolutionary pressures that may lead to some of these variants being successful. Some of them will be neutral, and some of them, like in this case with the child, will be deleterious and may compromise the fitness of the subject that carries the mutation. When we put together all the rare variants observed, and in this case is another study I completed that was published 6 months ago on 13,000 people, and I asked the question, how many genes can be truncated given that there is so many rare variants? And this will be the green line on the left. So the green line on the left is the number of genes that I observe truncated as we observe more and more individuals. And you can see that the green line keeps increasing, and the yellow line are a subset of those that are really particularly severe to the gene that carries them. That means that we expect that there will be a profound damage to the protein. What you can see is that as we go on, we will observe over 80% of the human genome, human proteome, can be observed in somebody as carrying a truncation of that protein. So it is like a human experiment of seeing how many genes can we knock out at least one copy and still be able to make a healthy, or we don't know if healthy, but a living adult. And you can see that we can knock out many genes, at least one of the two alleles, and still make it to adult life. Now, what you observed also towards the end of the curve, as we increase the number of variants observed, we do not reach 100%. That means, yes, there are genes that will not be observed ever with a truncation, even in the context of heterozygous, even in the context of being only one of the two copies. And this is what we call the haploinsufficient genome. That means those genes that you need both competent, complete copies to make a healthy adult or being born. So obviously this is showing you that for many genes, missing one copy will probably lead to embryonic lethality and not being observed in an adult population. This same data can be observed in children, in mice. You can also observe this phenomenon of the haploinsufficient genome and the fact that rare heterozygous truncations may be bad enough to produce embryonic lethality, and there's now plenty examples of rare truncated variants causing disorders in adults. So what can we do once that we have so much data? And I'm going to give 3 examples. You can opt for profiling individual risk, and here what we frankly call the wheel of fortune will be one individual, multiple traits, many of them are medical common traits, and you can produce what is called risk scores, that these are weighted or additive scores in putting together common variation and increasingly rare variants to calculate the individualized risk in a population. In this case, this person, for all the traits that we are computing, that goes from respiratory, neurological, or even phenotypic traits, you can see that for the most part, this person is, and I will put an arrow, this person is for the most part aligned to the average of the population. So it does not make a particular risk beyond the population average risk. Let's say average risk for asthma. You can see other examples where the person is high for some parameter. In this case, is some predisposition to melanoma, and in this case, this person has already had a melanoma. Much of this risk is calculated in this case from common alleles that reflect on the quality of the skin in terms of being pale and being more susceptible to UV light damage. Other cases, we do have what is called inherited cancer risks that are alleles with a bit more individual impact on cancer predisposition. You can also see that this person has some increased risk for Parkinson and Alzheimer's, and you can observe on traits that this person can also have some risk in terms of a trait like baldness, and this person is prematurely bald. This profiling individual risk is still very controversial. This is particularly powerful at population level. Many of the components are coming from large GWAS study, but increasingly we are able to include rare variants and therefore make even more individualized predictions of particular risks. Another generic application that you can and you will increasingly be seeing is the fact that the genome is providing more and more accurate information on traits. In this case, I can present here the prediction of a human face that includes not only the parameters of the face, the nose, most of them driven by ancestry, but also how you can estimate from the genome BMI, and you can estimate from the genome age, and therefore there are now tools that are beginning to give some prediction and some predictive value for forensics in terms of predicting the person's face. And it will be, for example, the face that here in the middle, if I can put the arrow. So if you get that face, actually we can predict the face, but we can actually predict age and weight from the genome, and therefore produce a rendition of the face by an artist, by program, that will try to adjust the face frame to the also genomically predicted age and BMI. Color, skin color, hair color, height, those are also pretty well predicted, and you can complete a pretty interesting, I would say, still not accurate, but very interesting prediction of individual remains from an accident or from a terrorist attack or from other forensic uses. You can reduce important aspects of the people. This, of course, opens a whole new discussion of the possibility or not to de-identify the genome. When genomes are beginning to be precise in terms of predicting biometrics, then you have to start wondering whether the concept of de-identifying genomes will hold true for long. So the genomics is becoming a very powerful genetic biometric tool. Now, what happens with rare variations? And I already touched on it when I was mentioning that one, there are lots of them in each one of us. Second, they are potentially new and therefore not tested for fitness. And third, they may carry a particular effect on a trait or a disease at any time through age. And this can be also happening, as I showed from the Haploinsufficient genomes, can also happen also from a heterozygous state of some of the rare variations with damaging effects. So let's take an example. This is a real data from the CDC. That's a normal flu epidemic. In this case, it was 2009. And what was, for me, very interesting was to compute the lethality of this epidemic on children. And even though it's a normal flu epidemic, you still find that 1,280 children died across the United States. And 40% of those, or around 500 children, were actually theoretically fully healthy. So you can think of it in Mendelian terms and say, 1,280 children out of 20 million, that's a 1 in 40,000, which is more or less a pretty common frequency prevalence for many rare disorders. So you can ask a completely fair question: is something as common as dying from flu or having a severe flu, could this be a rare disorder, a manifestation of genetics, and not just, like many people say, well, it was bad luck, it was a bad infection, and the child died? Says, well, let's think about it in terms of genetics. Can 1 in 40,000 represent mutations? And yes, you have exactly the scenario that we described at the genome level, that either from the novo or from rarity and Mendelian possibilities obtained from father and mother, you can have events like in this child, in this case of a compound heterozygosis from father and from mother, and I will put an arrow right now. So you can have from mother, from father, a compound heterozygosis that will produce a full damage to a particular important gene and pathway of response to a viral infection, that is the interferon pathway. And with other colleagues, we have now begun mapping many of the children to other genes in the interferon pathway, making true what I just stated in the prior hypothesis slide. That means that anything that is rare, anything that happens before the reproductive age, or even anything that shortens our lifespan even as an adult, can be analyzed under the concept of a genetic basis, even if it's a common disorder, even if there's no notion ever of this being a Mendelian. The fact, the observation, and the de novo possibilities of creating human variants, some of which are profoundly damaging, have to be considered as a fair hypothesis to pursue. So again, think genomics when you observe something that happens to a child before reproductive age, or something that is happening to an adult that will shorten the effective lifespan of a human being. And the effective lifespan of a human being now is somewhere between 80 and 90 years old. So dying at 40 years old from even a common disorder, you can prepare and pose the fair question of, is this genetic? Is having a tumor, a cancer at 40 years old, is it genetic? And there is a good possibility that there will be a genetic basis for an unexpected event. And I want to complete the next 5-7 minutes on another example of what it means to investigate the rare variation in humans. And in this case, I'm going to put not so much an epidemiological example, but I'm going to show you a functional example of how we are going to go through these observations of human diversity and its functionality. So here we have a particular cell, it's a dendritic cell, and you can see that there are green dots. I'm going to put an arrow. There are green dots right there in the middle. Those are viral particles being concentrated by this dendritic cell to do an important function, that is cross-presentation. That means the dendritic cell will exhibit and present these viral particles to a CD4, CD8 glial cell. Okay, so let's have an example. When you go through a database, you find that there are a fair number of humans. It's a very rare variant, but you have a fair number of humans that have a very early stop in this protein called Siglecl1. This is indicated there by the stop sign. I'm going to mark it. So many humans, not many humans, some humans, about 1 every 2,000, will have homozygosis for this particular Siglecl1. Obviously, they are alive. Obviously, they are, for the most part, healthy. And you can find them in exact, and you can find them in any repository at types, because this is not collected by genotyping arrays. This has to be specifically sequenced to be observed. But the fact is that there are humans walking around without this gene. So those that work in the antiviral and the viral pathogenesis field were surprised to learn that this important protein that exerts this feature of capturing and cross-presenting these viruses to the immune system, they were surprised that this person could be existing without this particular innate immunity protein. Okay, so let's move to the next slide. So the first question was, is this true? Do these people really lack this particular function? And here on the panel to the left, I'm going to mark it. You can see that's the density of this protein in the surface of people with a frequent allele, the common allele. This is in heterozygous, and this will be in homozygous. So what you can see is that there is no doubt people that are alive with a truncation of Siglecl1, they don't have this protein in the surface. There is no compensation possible. And even heterozygous are haploinsufficient. That means they have less protein than the common allele. So this protein exists. This protein is functionally missing when there is a truncation. And then we go to the biological assays. And you can see that anything that it is, now I'm going to mark it again, any particular parameter of function will be damaged as you try to do functional assays. And with viruses, in this case, it's a type of viruses based on both an HIV in what is called a VLP. That means it's covered with an envelope from another virus. And what you can see is this drop from dark blue to light blue. That means that the person that has a Siglecl1 truncation has no Siglecl in the surface, and this function cannot be supplemented by anybody else. Effectively, the dendritic cell loses the possibility to trap this virus and loses the possibility to bring this virus to the attention of the immune system. So once that this is done, you can and you will be persuaded that people with this particular allele will suffer, for example, HIV infection, the AIDS virus, differently, because you are seeing that the function has been clearly lost. Now, is that true? And when you go to the actual biology that we just said, that means that we lose Siglecl, we lose the possibility to trap and pass cells to the CD4s. And actually, when we go to big databases of HIV-infected people, we find that there are homozygous people that are infected, and I'm going to mark them here. There's people that are infected even though they lack the gene. There are people here, heterozygous and homozygous, they progress through disease. This is the typical evolution of an HIV person, progression through years and decreasing what is called the CD4 T cell count. That means the cell that is targeted by the viruses. And you can see that there is no difference between the evolution of people who are heterozygous. There are no difference in the difference of the possibility of developing profound disease on the homozygous. Here at the end, there's only 2 people. And also against the population averages, there is no evidence that the HIV-infected population has any modification of the allele frequencies, implying that they would be protected from acquiring the infection, and or that there will be any presence of a depletion, a particular truncated homozygosity in the population. So summarizing this slide means that even though the truncation exists in only few individuals, even though that truncation really impairs the presentation of this protein in the appropriate cell, the dendritic cells, even though the cell stops trapping the viruses and presenting it to the innate immunity system, that does not mean that in vivo that function is relevant. And here's where we start seeing the biology in the true in vivo setting, where wherever we think and we test in vitro, still we have to be confronted with genomic data. And I think this opens a new era where truncations will be, or damaging variants will have to be assessed not only in vitro functionally, but will have to be assessed at population level to really measure whether in vitro data truly translates into in vivo effects. So I have 2 more slides, and I will be stopping my part so you can make questions. One is that the example I showed for influenza, the example I showed for HIV, you can go ahead with many other truncations, like representing this particular panel of 8,000 individuals, and all those names on the top, we're going to mark them, like here. All these names are many viruses that affect the population, and you see that we have examples of truncating, severe truncated proteins for genes that are understood to be critically important for the containment of these viruses, at least in vitro. And I'm, of course, very excited to see that some of these viruses and some of these gene confrontations will be analyzed at population level to understand whether the in vitro evaluation of these proteins and these truncations will translate or not on population effects, like the one we saw with the child that got severe flu from a particular mutation. So we have many candidates in the population, and around you, many people will be carrying particular variants that will potentially put them at risk during childhood, during adulthood, during embryogenesis for a bad outcome. So I'm concluding there. This will be the summary of my presentation. The fact that we are now sequencing thousands of individuals is opening the door to understanding the extent of human mutagenesis. By now, we have observed 1 every 8th position in the genome has been observed by now variant. And so retain that number, 1 every 8th position in the genome has already been observed variant in some individual, usually through rare variants. So a large proportion of all human genetic variants discovered and to be discovered is individually rare. Maybe you are the only person that has that variant, but collectively there is millions and millions of rare variants now being identified across the human genome. As extreme example of this, we have now examples of human nulls and heterozygous truncations, and therefore the potential for haploinsufficiency or a large proportion of genes in the genome. And the current count is that probably 80 to 90% of the genes in the genome have been observed, at least in heterozygous truncation. And therefore the possibility for gene dose effect, haploinsufficiency, negative dominant effects. There are many ways that a truncated heterozygosity can do functional damage. And I put as examples severe diseases that follow either common infections or other common diseases, and you have to be attentive to think and push the genetic hypothesis, meaning that, as I mentioned earlier, the normal thing should be for a child to reach reproduction and from an adult to reach full species age, that means 80 to 90 years old. Anything that dramatically shortens either your childhood or shortens your adult life can be in fair terms ascribed to the possible genetic defect, because the material, the genetic substrate for such lottery, such possibilities exist, and we are now looking at it as we go through these large amounts of human genomes. I want to conclude there, and I'm happy to initiate the turn of questions and answers, and I'm passing the microphone back to Judy.
Excellent presentation, Dr. Telenti. Thanks for bringing that information to us. Before we get started with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. And our first question comes from Tammy. She asks, with respect to variants on exons, is it known the extent to which this impacts certain allele frequencies for certain genes?
So thanks, Tammy. Of course, there are several ways to answer to your question. One is all exons are subject of the same pattern of rare and common variation that I described at genome level. And if you go to the exact database and you do the same calculation, you will see that I think 1 every 8 position in exomes has been already observed with a variant. Now, the place in the exome will, of course, modify profoundly the allelic frequency because there are areas like splice acceptor, splice donor, protein domains that are critical, that will be extremely resilient to human variation. And when we go now through large-scale computing of the distribution of variants, you will clearly see the pattern of places where the exomes will be exempt from variation at some sites all across the genome. So not only we observed genes that may or may not be truncated, we also know across all proteins the positions that are uniquely protected from any particular damage, particularly splice acceptor donor sites. I hope that answers your question.
James asks, could all variants be placed into the context of energy? Sorry. Hang on one second. Sorry about that. jumped around a little there. Could all variants be placed into the context of energy-dependent changes in hydrogen atom transfer into DNA base pairs in solution and virus-driven energy theft, which causes mutations?
So thanks, James, for this question. I think, to be honest, I think we have to see every base pair change and its consequences. In this case, you are citing an interaction with a virus, very much in the biology and function of what is being happened. I don't think that summarizing all interactions on energy-dependent interactions will be appropriate for all mechanisms. It can certainly be appropriate for contact-dependent interactions, for example, surface interaction with receptors from viruses. All viruses, to a large extent, use human surface proteins for entry into cells, and they are, of course, very susceptible to energy modifications because these are the hydrophobic forces are part of the early contacts between cells. But once that you are at other levels, interaction with what we call antiviral genes or anything will depend on many other mechanisms that go beyond pure physical mechanisms like the one you described. Back to Judy.
Zara asks, which tool do you use for profiling individual risk?
So thanks to Sarah. There is a lot of interest in how do we compound and compute on more than one allele. What is called the genetic risk scores, like the one we use, represent an approximation that is probably not fully satisfactory to all of us of computing multiple alleles. So think about the system where a particular allele has been calibrated in a large population, 100,000 people, and you have an odds ratio, a risk ratio, a beta score from your logistic analysis that gives you a value. And this value may not necessarily be large. For example, a odds ratio 1.2 represents a 20% increase in risk. That same person may, at the same time, carry a protective allele with a 0.8 odds ratio, and therefore the computed risk for this person will be simply 0.8 added to 1.2, and you will give a neutral risk for this person. You can imagine that this is an amazing simplification of the problem, but to be honest, all the community is using this type of simple additive or weighted additive approaches to computing multiple alleles. And it works if you compute height. Height can be computed from 600 genetic variants, and you get a pretty good approximation to height, probably something like 5-6 centimeters differences in precision. So it works, but it's probably for those that love mathematics and statistics, is probably not very satisfactory. Back to Judy.
Okay, James asks, in substitutions near the receptor binding site determine major antigenic change during influenza virus evolution. The authors claim that the major antigenic changes of the influenza virus are primarily caused by a single amino acid near the receptor binding site. Do you agree?
So any interaction, again, I think we go back to the prior question we discussed. Yes, surface interactions between receptors and viruses are extremely susceptible to mutual variation that develops through human co-evolution with pathogens, in this case viruses, where the virus establishes a avoidance strategy, making mutations in the virus, and eventually the human being will have to fixate favorable alleles to counterattack a particular weakness of the entry site. This is referred in the evolutionary literature as the red queen effect, and it is the constant to and fro interactions between pathogens and human that force variants in both the host and the pathogen. So it's not one side or the other. Both of them will go through cycles of fixation and mutation to achieve the equilibrium in this ongoing interaction. Back to Judy.
Manju asks, which deficiency causes inflammation in humans?
So thanks, Manju. You are asking a wide question. There is an estimated, depending on conservative estimates or broad estimates, there is 400 to 1,400 genes that are labeled innate immunity in the human. Variants in subset of those are extremely well known to cause Mendelian disorders. Common variants in many of those are well known to produce susceptibility to more common disorders, including things like Crohn's disease, psoriasis, rheumatoid arthritis, celiac disease. So the question you are asking is absolutely correct, but it's extremely broad. It means every one of the 400 or 1,400 genes, depending how you count, can be observed variant, and many of those are being mapped to specific inflammatory disorders. And in many cases, these are really gene specific. So you may just knock out one particular gene from the interferon response, and you will only suffer a deficiency towards a particular pathogen, or you may change a particular set of innate immunity genes through common variants, and you will have a discrete increase in risk of Crohn's disease or of arthritis, and not other inflammatory diseases. So it is really a huge diversity of genes producing an important diversity of syndromes. Back to Judy.
Helen asks, what methods are you using to look at the contribution of rare variants of small effect to common human disease?
So Helen, you are putting there the finger in a difficult place. Yes, we are looking at particular models. One typical model that works in settings like, for example, mapping metabolome, metabolic changes. Metabolic changes typically map well to their genes, even through common variant analysis. And within those Manhattan plot type associations, at that area of association on the genome, we can identify multiple rare variants. They tend to have strong effects, so they add potentially powerfully to any score. But what we are not frequently confronted is on sums of rares. So typically, an individual with one rare variant may not carry a second rare variant, either to alleviate or to increase even further the mechanism. When that happens, we may go directly into real Mendelian disorders through compound heterozygosis mechanisms of dual rare variant effects. But it is a very exciting thing, and if you look at the literature, there is now real effort to try to push the two models, common variants, even low common variants, 1% to 5%, and rare variants into more personalized predictions. Back to Judy.
Olupade asks, how many of these gene defects can be associated with environmental influences, especially toxic environments?
So, Olapane, this is a very correct question. And let's go back to one of the models I presented when I said, well, you may be 40 years old and suffer from a cancer, a premature cancer. There is already evidence for some models where, of course, if you have a particular variant in a detoxifying pathway, if you have a particular variant in the DNA repair, and this is typical for many of the cancer predisposition genes, where you, like Lynch syndrome, where you damage the mechanism of integrity of the genome, any environmental influence will and may go unchecked. And that's where you may have a very important convergence between environment damage and genetic damage, where we can see that those two are together and not just individual effects on a thing. So you can say that, yes, you may have to smoke to have lung cancer, but you can also say that particular damages to your genome, correcting for DNA damage, correcting for detoxifying effects, will compound and increase the effect of the environmental toxic. Back to Judy.
Kavita asks, are we thinking in terms of SNV pathogenesis within genes?
So Kavita, if I understand your question, you are asking about single nucleotide variants pathogenesis, and I hope I understand well your question. Of course, there is single nucleotide variation that we are beginning to understand better, and huge effort on all other forms of genetic variant. Typically, individual will carry 4 million single nucleotide variants and maybe 200,000 very small indels, 1-2 bases, and then it will have some hundreds thousands of larger structural variation, some copy number variations, and some other forms of modification of the genome. Now, the reason I don't make currently too much emphasis is because there is still some precision issues on calling correctly copy number variation. And there is a huge interest of trying to get better colors of structural variation, copy number variation, other forms of diversity to analyze them in the context, again, of traits and diseases. Of course, there are many, many good examples of all these things causing human disease. And the only issue here is that the purely short read driven genome sequencing has some limitations in precision when calling copy number variation or in structural variations. Back to Judy.
Tammy asks, is there genetic redundancy of genes that are found to be null in some individuals? And are these found in certain biological pathways? Or are these only genes that function in immunity?
So thanks, Tammy. Your question is very appropriate. I think all of you are well knowledgeable in the fact that there is gene families, there is gene duplications, there is things like cytochromes where you have multiple times genes that are all a bit similar, and therefore there is an intense and important substrate in the human genome for redundancy. There is a lot of debate here whether we have pure redundancy, that means another gene can take over the loss function of a gene that has been lost, or whether this robustness, that means that the network is able to accommodate the loss of one of its members. and there are models for both systems. Some of the things I've showed to you in terms of haploinsufficiency taught us that many of the haploinsufficiency events will not be compensated in terms of the gene catching up to the appropriate levels of expression through the one single leftover copy. So we know that there is a lot of capacity of the network to be robust, and we are in some ways less convinced that you can speak about pure redundancy where another protein, another allele takes full compensation of the defect through the same pathway. So I hope that answers, and of course this is beyond innate immunity. Any system really lacks, has this property of robustness through redundancy and compensation and intimate robustness of the network. To Judy.
Okay, we have time for one last question, and it's from Bill. What do you mean by collectively prevalent? Are you referring to the variant or the individual?
So Bill, thanks for the clarification. Yes, so if you have 100 individuals and everybody has one rare variant, you can say every single individual has a rare variant, and therefore we term that collectively prevalent, but individually rare. So the variant is not the same one. Collectively, everybody is carrying rare variants. So I think it's just to misspell the doubt that people say, well, rare variants are things that happen rare in the population. Rare variants happen as a unit rare in the population, but rare variants are extremely common because everybody has tons of rare variants. That's what it means, collectively prevalent. And back to Judy.
I would like to once again thank Dr. Telenti for his presentation. Do you have any final comments?
No, just thank all the organizers. This is a great opportunity to present our work, and I'm really very impressed by the number of questions and the interest and the follow-up of this forum. Thank you very much again.
Thank you. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, and to get your CE credits, click on the CE button located in the bottom left-hand corner of your web page. Today's webcast will be available for on-demand viewing through November 12, 2016. You'll receive an email from us alerting you when it's available on demand and posted on labroots.com. You're welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. See you next time.